Okay, it is 2 o'clock. We're going to go ahead and get started. Welcome, everyone. Thank you so much for joining us today. This is a brand new series for us on scientific literacy, so we are super excited to introduce it to you, and thank you for being a part of this with us. I'm going to let our special guest, Brian Magnier, kind of go over the format of how these next 4 weeks will go, and I'll just do the housekeeping for us for now. We will keep our mics and our cameras off for this first session, but the next 3 will be more classroom style. And I do want to give a big thank you to our friends group. The Friends of Brooker Creek Preserve have graciously sponsored this series, so we thank them for supporting us in our efforts. And if you are interested in learning more about the Friends or becoming a member, you can check them out at thefriendsofbrookercreekpreserve.org. And I'm going to turn it right over to you, Brian, and we'll jump right in. Thank you so much for being here with us.
Awesome. Thank you, Julia. Yeah. So we're going to talk today and for the next few weeks about scientific literacy, and a little bit of media literacy as well. There's kind of some overlap there. Today, we're really going to kind of dive into scientific thinking. What is science? Kind of the essence of it. A little bit about probability and some statistics. Nothing too fancy. We're not going to need a math degree to understand today. We're going to kind of start from the ground and work our way up. That way we're all kind of on the same page. But a kind of an important part of understanding data and science and all of these different sort of bits of news that you hear, an important part of that is being able to put it into some sort of context with statistics and biases and likelihood, and all of this kind of revolves around just scientific literacy. Kind of is the little thing that we're kind of centered around for all of these little side topics. So with that, let's start just kind of defining what science is. The essence of science really is gathering observations of the universe, using logic and reasoning, math perhaps, to come up with patterns and rules for how things work. And science is both a process and a body of knowledge. If you think of the scientific method, that's kind of the process. That's one process by which we do science. But these things, they feed off of each other. Anyone can make observations about their environment. You don't need to be a scientist to make observations. Just like how even my cats, they've figured out what time they get fed. They definitely let me know if I'm late giving them dinner. They've made observations about their little universe. And they can see patterns of, what time of day they expect food. But once humans specifically developed language, and even more so writing, that's when science, I'd say, was really born. Because then for the first time, we could communicate and share our observations with each other. We could build up a framework of knowledge using other people's views, thoughts, and data. And ever since then, the process of observing the universe has been kind of adding to humanity's body of knowledge. And in turn, that body of knowledge influences how we perceive the world around us. So that's how I see these two things, the body of knowledge and the process, feeding off of each other in kind of this weird little symbiotic relationship. Now, the essence of scientific literacy specifically is just kind of the ability to be able to draw evidence-based conclusions about the universe, given some sort of information or data. So the tricky part, especially in today's high-speed, high-tech sort of world, is being able to parse through the mountains of information, especially misinformation, and all of the biases to find the truth, assuming, as I do, that we live in a kind of real, tangible universe with physical laws and knowable facts, rather than some simulation or a fantasy land that was created inorganically. If you assume that the universe has actual laws and truths to be known, there are facts out there. Science is the way that we can kind of get at those facts as accurately as possible. So one major distinction that we have to think about is where we are getting our information. Let's say if you're walking in the woods, directly observing some insects, let's say, and trying to understand their behavior. In that case, it's useful to know something about insect behavior, ecology, maybe evolution, to put your own observations into some sort of a framework so that you can make an informed hypothesis about what's going on. Why are these bugs behaving like they are? Or are you at the proverbial water cooler at the office, trying to make sense of some summary of a scientific discovery that your coworker is attempting to convey based on a short article that they skimmed over breakfast that morning? In that case, scientific literacy may even be more critical, since the information that you're receiving is being filtered and broken up, kind of like a game of telephone, from the original researchers to the media, to your coworker, and then to you. So you must rely on this combination of logic and prior knowledge to figure out what you should actually believe. And that's really what we're going to be talking about for the next 4 weeks, is figuring out what you should believe and why, or at least giving you the tools to make those decisions a little bit easier for you. So if your relative is telling you over the phone about some crazy new miracle food that cures all ailments, or maybe an article on your Facebook feed initially sounds too crazy to be true, that's when you want to try to think critically and skeptically. Maybe ask yourself these 3 questions here. Does the assertion make sense? Does it make logical sense to you? Let's say if somebody claims they have ESP and they can read minds, what's the mechanism by which that information is actually being transferred? Things still need to follow the laws of physics. So if somebody makes some sort of a claim, you want to have it make sense. And then you also need to have evidence or data that backs up that assertion. Could there be alternate explanations for those observations? If you see a picture posted online of a potential UFO sighting, consider the myriad other things that are more likely, even if they're more mundane, than an alien invasion of Earth. When a movie claims that a superhero can fly because of some genetic mutation, consider how genes actually alter organisms. DNA, it just codes for proteins, and proteins interact according to the rules of chemistry and physics. So genes are not some magical entities that give their hosts abilities to fly. They might code for wing bones or feathers or different muscle groups that together would allow the host of those genes to fly. But there's no single gene that lets somebody break the laws of physics, ignore gravity, and spontaneously fly. Now, I love Marvel movies as much as the next person, but just in terms of thinking scientifically, we have to remember that everything in the universe, as far as we have found so far, can be explained logically and methodically. And that's what we're going to be talking about. So here's a website that I visit often and definitely recommend to find science news. It's got more up-to-date discoveries and more accurate information than you'll generally find on your Facebook feed, sciencedaily.com. However, even at this website, you can't always escape misleading, over-sensationalized sort of headlines that pervade the internet. If you take a look at the article that the title that I circled here, how does it strike you? So here's one of the papers that this. So Science Daily. Let me back up. This website here, it takes peer-reviewed papers and then it kind of gives a little summary of them. So it doesn't have the actual original research and data here. This is just giving you little snippets of kind of the new discoveries, the highlights in science. And so this one here, the title is Humans' Ancestors Survived the Asteroid Impact That Killed the Dinosaurs. That sounds exciting, right? But does that make sense? Since humans exist, then obviously our ancestor survived that KT mass extinction, the asteroid that killed off the non-avian dinosaurs. So the title doesn't really mean anything, right? Like obviously our ancestors had to survive because we're still here. So if you actually click on this and read it more thoroughly, and especially if you follow the link to the original peer-reviewed article that it's derived from, you see that what it's actually talking about is that placental mammals, of which humans are a part of, they likely evolved before the asteroid hit. And so this group, the placental mammals, survived the asteroid. And so, this asteroid wiped out the non-avian dinosaurs, but not the placental mammals, which are our ancestors. It's the part of the group that we're in. But it's not exactly the same as saying that our ancestors survived the impact that killed the dinosaurs, because that's kind of an obvious thing since we still exist. But we'll talk about, sensational headlines and overreaching summaries more extensively next week, especially. For now, let's move on to some statistics. Statistics and cognitive biases, these are kind of more logical, mathematical ways of looking at the world that can hopefully, once you understand them, strip away some of those over sensationalized sort of bits and let you kind of take a more objective look at what's being presented. So most of you probably have a grasp of, basic probability theory, whether it's from a math class a while back, or maybe just from playing lots of blackjack and poker or any other games that involve chance. Here are some of the basics, just so we're kind of on the same page here. So the idea is the probability of something happening is equal to the number of desirable outcomes, whatever we say is kind of this event happening, divided by the total number of possible outcomes. And so that's just kind of a fancy way of saying, let's say we roll a standard 6-sided die, and there's a 1/6 chance of rolling a 2 on our first roll. There's a 1/6 chance of getting any specific number because there's 6 options. If we pull a card at random from a standard 52 card deck, there's a 1/4 chance that it'll be a spade, since there are 4 suits to choose from, 13 of each. There's a 1 in 52 chance that you'd get exactly the ace of spades by pulling a card at random. This is kind of the basic foundation of probability theory. So if we do, another common way that people look at and understand probabilities is with coin flips, because these are, they have 2 outcomes, heads or tails, nice, easy 50-50. But let's pretend you've never seen a coin before. And you just want to investigate the properties of this little weird metal circle that you've never seen. And the way that you can interact with this little object is you can flip it and you can make an observation. So you come in with no knowledge of statistics, no biases, preconceived ideas, none of that. We're just going to take a coin, flip it in the air, let it drop, and make an observation. So there's about 20 of us here today. So let's say the first flip, everybody sees something. You see either a head or a tail. Everybody sees that one thing. Now, let's say you flip it again. Half of us will have only seen either heads or tails. We've gotten 2 heads or 2 tails. The other half will have seen both sides of the coin already. But we can keep doing this successively. And after 3 flips, maybe a 1/4 of us have only still seen one side of that coin. And after 4 or 5 flips, the chances are at least one of us in this group that are here online right now, at least one of us will probably have only seen just heads for 4 or 5 flips in a row, or just tails for 4 or 5 flips in a row. And so that's kind of this little analogy for understanding kind of how science works. If you make more and more observations, you're more likely to get at the truth, but there's still an element of chance in there that can kind of screw you up. Because if you didn't know what a coin was and you didn't know that there are heads and tails and things like that, a percentage of you would see this coin and you flip it 4 times in a row and it only comes up heads, and you'd say, this little object, when you flip it, it always looks like it always comes up heads. And if you were to stop flipping, you would think that object always came up heads no matter what, because now you've got this preconceived idea of every time I've done it in the past, it's come up heads. So in the future, it'll probably only come up heads too. And that's basically a way of saying that, if we do scientific experiments, there's some chance involved that maybe the scientific experiment will come up one way, when even though that's not actually how, it doesn't reflect reality, let's say. And so you kind of want experiments to be repeatable, and you want to have good, solid sample sizes and statistics to back up any assertions that you're going to make about the universe. So let's look at a couple different types of biases that can kind of skew how we view the world and data around us and things like that. One common example that people talk about is called survivorship bias. And here I've got a plane. Why do I have this plane with a bunch of red dots? Well, kind of a classic example of survivorship bias was back when, World War I, World War II, you've got these planes flying around. I think this would be World War I here with this type of plane. We've got, Anyway, let's say the planes are going out and battling and they're shooting at each other, and a certain number of planes come back, and the engineers that are trying to fix these planes, they see where all the bullet holes on the planes are. You know, these planes have been out in battle, they come back home and they see, wow, these planes are always getting hit in the wingtips, in the tail tips, and kind of right in the center mass. So that's where we want to put a bunch of armor, right? That's where all the bullets are hitting. So let's cover up those spots to make sure that next time this plane goes out, it's protected from those bullets. Does that make sense, right? You know, we're seeing where the damage is done and we're protecting those spots. Now, the problem with this is something called survivorship bias, where the planes that come back, they're the ones that got hit in places that weren't actually deadly to that plane, right? It didn't get hit in the engines or in the cockpit where the pilot is. It didn't get hit kind of in the middle of the tail that would cause the whole tail to fall off. So these are actually hits that are okay, that the plane can sustain. And so when you're trying to shore up all of these bullet holes and protect the places that got hit, you're actually kind of doing the opposite of what you want to, because the bias of which planes came back is the ones that survived. Ideally, you'd go look at the planes that got hit and fell down and got taken out of the battle, and you'd look at where those guys got hit, and that's where you actually want to protect. Let's take a more naturey example, you know, get back to the fun little critters. So you can think of an example of survivorship bias when you see leucistic or albino birds, right? Because these guys exist. There's some weird genetic mutation that affects the pigment production in different organisms, and it might shut off the certain pigment production, and so they look more white. Like these here, we've got a killdeer and a chickadee that look more white than they should because they're leucistic. They've got these mutations that affect pigment. Now, in general, the leucism rate in birds is about 1%, and the albinism rate is about 0.01%. So very few birds are leucistic or albino. But even that, 1%, that's actually pretty high, because if we look at a big flock of birds, look at this, there's more than 100 starlings in here. And if 1% of them were actually leucistic, you'd think some of these guys would look pale or whitish, right? Well, the survivorship bias might be taking an effect, might be an effect here, because maybe the ones that are white are eaten by predators because they stand out in the flock. And so even though maybe at birth or when hatching, 1% of those starlings, say, are whitish, by the time they're all adults and flying around, those ones have kind of been picked out of the herd. And so what we're left with is a much larger number of normal, non-mutated sort of adults. And so it looks like the rate of leucism and albinism is much lower than it actually is. On the flip side, we could go back and we could say, where did they get this number? You know, do we trust this number? Do we trust that these researchers got these rates right? Maybe, you know, when whoever found these rates, maybe they had kind of just an abnormally high number of mutant birds in their study, and these are actually not the right numbers at all. So that's kind of, you know, there's two ways to look at how many birds in a flock might be albino or leucistic. Are we seeing survivorship bias taking out those albino birds, or are we seeing just the initial assumption being false? Okay. Another major bias that comes into play is called sampling bias. This one here is kind of a bias just in terms of where and when we are looking at the universe. You know, let's say you're going to look for whales, you know, and you go to the coast, you go up as high as you can. I guess in Florida, you can't get very high up off the water, but let's say, you know, you go up to a little bluff or maybe up on a lighthouse or something, take your binoculars and you scan and you're looking for whales. And, you know, that's where you're going to see whales, is if you're on the coast, right? You're not going to see whales if you are right in the middle of Florida or inland in the mainland US. And so we don't see whales on land, but we see them when we're right along the coast there. And so they're all reported right along the coast. You know, see almost all these orange dots here. This is on iNaturalist. Almost all these orange dots here are reports and pictures of cetaceans, whales and dolphins. But does that reflect where the whales and dolphins actually live? because I'm pretty sure you'd probably find whales and dolphins out here in the middle of the ocean. They don't just hug the coast. That's just where we are. That's where we're making our observations, right? So it's a lot more likely that they live all over the ocean, but the sampling bias is making it look like the whales only live right on the coast, because that's the only place that we're able to actually see them. That's the only place we're making samples. That's the only place we're making observations. And so this is an example of sampling bias. Another good example from Florida would be looking at eBird data and kind of seeing where different birds are seen, how many birds are seen in different places. So here's a nice local spot for many people on this call, hopefully. We've got Honeymoon Island and we've got Calidesi Island State Park. Both of these spots are these little, sort of barrier islands off the west coast, the Gulf Coast of Florida. They're very similar in terms of area, habitats. They've got these different little sort of salt marshy areas. They've got beaches, some little stands of trees for migrants. And so you'd expect them to have very similar types and numbers of birds fly through. But instead we see, if we look at the eBird data, Honeymoon Island has over 100 more species seen than Calidesi Island, even though they're right next door. Now you could try and explain that with different habitat type things, and maybe Honeymoon Island just has lots of good food and the birds like it more. But I think, if you take this extra little bit of data this extra bit of information into account, it might make more sense. Honeymoon Island is connected to the mainland by a bridge, and it's a very popular park. A lot of people go there. Calidesi Island, you need to have a boat to go explore it. It's not connected to the mainland. You can't just go there and drive over there for a little picnic. You kind of need to make some effort if you want to go explore that island. And so with that being said, it makes sense. All the birders are on Honeymoon Island. All the observations are happening on Honeymoon Island. And so the fact that it looks like Calidesi Island has way fewer species might not actually be a reflection of the reality that the biodiversity that the two islands have. It's more likely that, we just don't have the people going there. And if we had as many people going to Calidesi Island on a daily basis, eventually we'd probably see over 300 species of birds there. And I kind of have a personal story, a personal connection to this sort of sampling bias and a little bit of what we call confirmation bias that I'll mention in a second. I wanted to see dusky pygmy rattlesnakes. This is a few years back. This is my discovery of Brooker Creek Park for myself. I wanted to see dusky pygmy rattlesnakes around Tampa. And so I went on to iNaturalist and I was like, okay, where's everybody been seeing these pygmy rattlesnakes? And so I found a nice cluster of sightings at Brooker Creek. I was like, oh, that must be a great place to go find these snakes. So I went there and sure enough, I found them and it was awesome. I got to get pictures of them. It's a great place, Brooker Creek, the trails. It's a great place to find these dusky pygmy rattlesnakes. But it's kind of a bit of confirmation bias, right? I went to a place and I saw the thing, and now I always go back. If I want to see dusky pygmy rattlesnakes, that's where I go. If I want to show somebody a rattlesnake, that's the place I always go. And so I'm not really sampling other places equally to see if maybe there's plenty of rattlesnakes over here at John Chestnut's, at Chestnut Park over here. Maybe there's plenty of rattlesnakes over there, but because there's no sightings there, I'm not taking the time to go look for them there. And so the more sightings you get in one spot, the more people go to look for those critters, the more sightings are going to be made. And so here it's kind of a combination of sampling bias and confirmation bias, because I'm only sampling one spot. I'm really only sampling Brooker Creek. And then every time I see a pygmy rattlesnake there, it's this confirmation bias. Oh yeah, good. Brooker Creek is the place to see these guys. And if I really wanted to know where the pygmy rattlesnakes are most common, I should go to different parks every weekend. I should go to a different park. And then, after a year of sampling all these different places, I should see which places actually had the most pygmy rattlesnakes. And that might actually give me a better sense of where they're actually distributed. But I know that they do exist at Brooker Creek because of this sampling bias. And so that's kind of where I always go. That's in kind of the same line of thinking. There are these different fallacies, these different sort of logical problems that that have to do with your own sense of probabilities and likelihoods of seeing things. And so you might have heard people say, oh, you're more likely to be hit by a car than to be struck by lightning. And that's true in general. But if you're out in the woods, walking around, or you're in like a big open field, walking around during a thunderstorm and there's no roads nearby, then that little bit, the fact that you're more likely to be hit by a car than struck by lightning, that doesn't really give you much reassurance. That might not actually be accurate once you're out there away from roads and in the middle of a thunderstorm. And kind of the same thing, people will often say, oh yeah, most shark attacks happen in less than 3 feet of water. Just like with the whale observations, that's just where all the people are, right? That's a major sampling bias. And so you can kind of start to see these things where people will say some sort of a statistic or a fact, and that may be true in general, but you have to remember to put it into a context. There's different conditions. That's why we call this conditional probability here. We've got the probability in general might be more likely that a human is more likely to die by car than lightning, but a human on a mountaintop in a thunderstorm is probably it's almost impossible for them to be hit by a car. It's probably a lot more likely that they're going to be struck by lightning. And so you have to take different conditions into account. They kind of shade, these different statistics that are reported. And I know we're jumping around a little bit. We're trying to cover a lot here just to kind of lay the groundwork for some logical thinking and some statistics. Let's have one more kind of big example of a type of statistics that's going to be useful in kind of understanding science. It's called Bayesian statistics. We're going to look at Bayes' theorem. Don't worry, I know, big variables, big old equation here. We're going to get into it. You're going to understand it in just a few minutes, I promise. It looks kind of daunting, but we're going to use 2 examples that I think will make sense. And then we'll understand what all these letters mean here. So the first example is disease testing. So we're talking about Bayes' theorem, Bayesian statistics now. So you go to the doctor for a routine blood test, and when the results come back, you've tested positive for some terrible disease. You don't want it. The doctor says that the test has an accuracy of 99%. What are the chances that you've got this disease? Well, you might think 99%, that's the accuracy of the test. But that doesn't actually take into account all the other information, all the other conditions that are floating around. How rare is the disease? What's the frequency of that disease in the population at large? So let's say the terrible disease afflicts only 0.1% of the population in general. Does that extra knowledge change how we view those test results? Well, we can kind of flesh out this example here. Let's take about 1,000 people. Got a whole bunch of people here, big sample size. Now let's give all of them that kind of just general blood test. So we expect 0.1% of the population, or about 1 person in 1,000, to have this bad disease. They're that red person down there in the corner. We would expect the test to accurately recognize them 99% of the time. But if we're testing 1,000 people and the test is 99% accurate, then 1% of the people tested will be false positives. So in our population of 1,000 individuals being tested, we would expect the test to show up, to show that 10 people have this disease, right? Just even though they don't have the disease, the test is 99% accurate. That 1% inaccuracy, if you apply it to a large number of people, it's going to incorrectly identify a lot of those people, about 10 of them here. So in our population of 1,000, all of whom get random blood tests, we expect 1 person to actually have the disease. 10 of them are going to be false positives. So if you're in this group of 11 who tested positive for the disease, your chances of having the disease are actually only 1 in 11, or 9%, not 99%. So that bodes well for you, right? You went to the doctor, you got this blood test, you were all scared because it said that you had some horrible disease. You've actually still got a pretty small chance of having the disease. So we can use this example here to understand this kind of ugly looking equation. So what does this mean? So what we've got here is Bayes' theorem, and let's walk through it step by step using this example that we just kind of walked our way through. So we've got the probability of having the disease. So on the left side of the equation, we've got P is probability. The probability of A, the probability of A is the probability of having the disease. Probability of B is that we tested positive. So the probability of having the disease, given that we tested positive, so B is a positive test result. So that equals the probability that we tested positive, given that we actually have the disease, that's that one red person, times the probability that we actually have the disease, and that's 0.1% probably in this population, divided by the probability of testing positive for the disease, because that's all of the options. We went to the doctor and we know we tested positive for the disease. So if we add all of our numbers in here, we've got our probability of having the disease, given that we tested positive, that's this little left bit, is going to equal 99%, because that's how accurate the test is, times 0.001%. That's how many people in the general population have this bad disease, divided by the fact that, so 99% of the people that have the disease will be identified correctly, and 0.01% of the people will be misidentified. So most of the population, 0.999% of the population, don't have the disease, but they'll be incorrectly identified. And so if we work through this equation, got all these numbers, big scary numbers, but we just kind of whittle it down, we get 9%. And that matches what we had when we looked at the big giant, group of people. And so here now we kind of are starting to understand this equation. Maybe it still doesn't make sense exactly. We don't know what all the variables exactly mean, but now we've got this sense, if you look statistically and mathematically, even if you test positive for this disease, we have to know something else. We have to know more information to actually make an accurate sort of assessment of what's going down. So let's try and do a nature example. Let's say you're driving around in Florida and you see what you're pretty sure is some big falcon, but it's pure white. This amazing picture of a gyrfalcon. It's actually a peregrine falcon. But let's say you're driving around Florida, you see this big, chunky sort of white predator bird sitting in a field somewhere, and you think, wow, this is Florida's first record ever of a gyrfalcon. They're usually far north, usually in northern Canada, Alaska, Russia. This would be an amazing bird to see in Florida. Basically, nobody sees this. Nobody has ever seen this bird in Florida in the wild. But what's the likelihood that that bird is actually in Florida? One of those individuals, a gyrfalcon, would have had to have flown maybe 2,000 miles south off course. So it's probably a very small likelihood that actually happened. It's kind of like the likelihood of having that bad disease, right? Very small chance of the gyrfalcon actually being in Florida. The things that might be more likely are maybe we just saw a different, more common bird, and we just kind of saw it wrong. Maybe we saw it in kind of bright light, and the belly of the red-shouldered hawk here, the short-eared owl, maybe they just looked a little bit too white. And so we kind of mistakenly thought that it was this gyrfalcon. Or maybe even they're albino. Maybe it's more likely to see an albino red-shouldered hawk in Florida than it is to see a white-faced gyrfalcon in Florida. Other options, other ways we could interpret kind of this observation. Maybe it was just a snow goose at a weird angle. Maybe we were driving by on the highway. You only see this thing for a split second. You thought it looked like a falcon, but it was actually just, a different type of white bird. Or, and I'm very guilty of this, maybe it wasn't even a white bird at all. Maybe it was a plastic bag in a field, So there's a lot of different options that you have to take into account. So if your coworker says, oh, they saw this white gyrfalcon on the way to work today, they Googled some pictures. The white gyrfalcon is what it looked like, to their memory. Trying to, if you calm down a little bit, don't rush out and try and find their gyrfalcon immediately. Maybe take a second and think, what's the likelihood that actually happened? And maybe it's more likely that what they saw was, one of these other probably more likely options. let's see here. Okay. So kind of changing pace a little bit, just again, thinking about initial impressions. First we thought we saw a gyrfalcon, turns out it was something else. One interesting example that I like to kind of bring up when thinking about initial impressions and sort of different biases coming in is when people look at whales and they try to classify them and they think, oh, that looks like a fish, right? It can be very difficult to discern what's just convergent evolution, organisms looking like each other, and what's actually relatedness. And it's interesting how common it is for people to think of whales as fish or mistake them as fish, because scientists for the last 2,000 years, since the days of Aristotle, they've known that whales are not actually part of the group that we call fish. They've known that whales are something separate. And for hundreds of years, we've known that they've been mammals. But many people, especially kids, they still kind of conflate those two groups because you're often looking at superficial characteristics or brief sightings. These are just creatures that live underwater, ergo they must be fish. And it wasn't until recently, about 60 years ago, when scientific sort of taxonomists actually proposed that whales evolved from terrestrial ungulates, hoofed mammals like deer. So that's kind of weird that whales are evolved from these terrestrial mammals. But it's just this example of, if you have these initial impressions of what a thing is and you try and categorize it, it can take some effort. It can take some science, some scientific methods, some research to actually figure out the deeper truth behind it. The fact that whales are not actually fish. Kind of the same sort of thing is if you've ever seen these weird little plants in the woods, a lot of times you think they're fungi at first because they're not green, they've got no chlorophyll, they like the shade. That seems like kind of a fungusy thing to do, not a plant thing to do. But if we look deeper, if we look at their cellular structures, we look into their genes, their DNA, we can see these are actually plants. They've just kind of gone the more mushroomy route of survival. They don't have chlorophyll. They have these little roots that steal nutrients from neighboring plants and things and fungi. And so they act like fungi, but they're still plants. Okay, I've thrown a lot at you. Let's kind of take a deep breath. We understand some potential biases. We've looked at a little bit of statistics. You don't have to remember all of it, and I can always go back to any of the slides at the end if there's questions about specifics. But let's just say we kind of are now, our brains are geared to thinking about these biases and these statistics. So now let's go outside and we make some observations. We diligently record what we see. Maybe we witness some interesting behavior that we don't know a lot about. Get back home, we hop on Google Scholar and we try and find this behavior. Who's reported this behavior? What's it about? Why are these scorpion flies flashing their wings? Why are they waving their wings around? Is that some sort of communication thing? Is it just like a stretch? Some physiological response? We don't know what's going on. But maybe we can't find any information on it. Our research doesn't turn up much. Maybe that behavior is cool enough. We want to write it up. We want other people to know about it. So let's go back outside, make more observations, take pictures, take videos, things, taking in data so that other people can see the same thing that we're seeing. They're not just going to take our word for it that these scorpion flies are doing some sort of a mating dance or maybe some sort of territorial display. We have to try and prove it. We have to try and prove all the other options are less likely. And what's actually happening is these guys are doing some weird territorial display that hasn't been described before. So that's how you kind of take some of the theory. We're going to do some statistics. We're going to make sure we look at our own biases. We're going to take that and we're going to apply it to the world around us. But a lot of times we're not the ones out in the field, out in the woods doing the observations, right? I think a lot of times we're sitting at home, we're watching the news, we're scrolling online, and that's when we're getting our information from. And so what we're doing in that case is we're taking taking words from other people, from scientists and from kind of intermediate media people who write popular science articles. We're kind of taking their information and then we have to process that. But we still have to do the same sort of scientific critical thinking. We still need that same scientific literacy, right? We still want to look at the data and understand what's actually going on. And we want to decide if we actually believe it, right? Especially if we're just kind of looking at little headlines, maybe even memes on Facebook. We want to understand whether we should believe a source or not. So let's kind of just take a look at this process. It helps to understand what's going on behind the scenes before you see information pop up on your Facebook feed, let's say. So scientists are going out, they're observing, collecting, and recording raw data. Maybe that's taking those pictures of scorpion flies flapping their wings around. They analyze the data, they summarize it into an academic paper. That paper may get picked up by a popular science news outlet, maybe a magazine, might be further summarized, put into some sort of context with other research to make it just more readable, get rid of all the statistics and the numbers, just kind of summarize what's happening. Maybe the scorpion flies are dancing to protect their territories, and that might be a summary of what the data says. Those popular articles then might be repeated by other outlets, other individuals, maybe in the form of, just little Facebook articles, YouTube videos. And then people see those and they repeat that information to other people, to family and friends. But by the time you hear it, like I mentioned before, there's this game of telephone going on, and there's so many different ways that information could be distorted or misrepresented. And these red examples, these are all kind of, even when science is happening normally with no malicious intent, this is just errors that we really can't completely control for. We can be aware of them, but it's very difficult to eliminate them completely. So even right when the scientists are observing things, they've got biases, they make mistakes, they might cut corners because of funding or time. Maybe with the scorpion fly wing waving thing, maybe they do something totally different at night, but the scientists weren't out there at night to observe it, and so they're not getting the whole picture. Maybe when they analyze the data, just random chance kind of skewed what the statistics looked like, and so some of the data was a little bit misrepresented even in the original paper. And then when popular science outlets pick up articles like this, they pick and choose the weirdest and the wackiest and the most strange, outlandish sort of science, right? Because that's what people want to read. People want to read the new discoveries, the cutting edge stuff, the weird headlines. And so there's this major bias toward odd and weird findings. And then that happens again, that gets compounded in things that go viral on Facebook. The weird findings, the unexpected results, those are the ones that people will repeat even more, which means they're going to get summarized and paraphrased even more. And often these popular science articles, they're kind of removed from the peer-reviewed science, and they might be written by people that don't fully understand science or statistics. Might be written by just, writers who just want to get content out there. And so they might misconstrue accidentally or intentionally kind of the true nature of what's going on with the original observations, with what's happening in nature. And then, of course, the last step in this game of telephone is that, you read an article, maybe it's bedtime and you're just reading to fall asleep. And then the next day you repeat it and you kind of half remember all the information. And so misinformation is kind of spawned that way. And that doesn't even take into account, I know this is looking kind of bad for how much we can trust science and everything that we read online, but that doesn't even take into account initially, maybe scientists were funded by people who have some sort of agenda. And then also people up on the other end, people are writing, just little Facebook articles or memes. They might intentionally mislead or over sensationalize headlines just to get more clicks, right? So they might not care about what the content says. They might try to write some sort of headline that's just going to grab your attention and make you click more often. And that is kind of what happened with this article that I've noticed on Facebook. So one headline that keeps making the rounds on social media, and maybe you've seen it too, I've seen it a few times, is this here. It says that an extinct bird re-evolved itself back into existence. It says that, the bird went extinct, and then 20,000 years later, it came back and evolved again. And I've seen this actually, in meme form. This is one article that has kind of broken through even just popular science circulation. And now it's all these different memes. It's saying, this bird re-evolved itself. It's, oh, good job, we're no longer extinct. It's a lot of different funny little memes around this sort of article. But if we actually go to the core of it and take a look at this article, does that make sense? What does that title mean? What evidence is there for this assertion? And that is what we're going to look at next week, because what we're going to do, Julia will send out 2 PDFs, both the Popular Science article, which is this one that kind of keeps making the rounds, which is very short, pretty easy to read, pretty easy to digest, for better or worse. She's going to send out that article, and then also the original peer-reviewed academic sort of paper, a PDF of that. And that's what the Popular Science article was derived from. And so what we're going to do next week, hopefully before then, you can read those 2 papers, because Julia will email them to you, or post them somewhere for you to download. But yeah, you can read both of those. And of course, the academic one's a bit longer. It's a bit drier. I don't expect you to necessarily read and understand every single word, but that's what we'll talk about next week. And what we're really going to try and dive into next week is that headline true? Does that make sense? And then also kind of look at some of the other, the big leaps in terms of going from the actual data to what gets reported and talked about in kind of more general social media, say. So if you see an article that sounds amazing and sensationalized, let's follow it back to the original data and what evidence are we actually using to support that claim. And so that's really what we're going to try and dive into next week. And I'll have a presentation as well. I've got a bunch of slides, but we don't need to get through all of them because I want to hear from you guys too. And so next week we'll have cameras and audio on, or at the very least, we'll be able to put stuff in the chat, perhaps. And, we'll be able to kind of talk a little bit more back and forth about your interpretations of these 2 articles. So hopefully before then, you take a glance at both of them. And then the following weeks, so on the 19th and the 26th, we will be talking about these other 2 papers that Julia will also share with you guys. And we'll be kind of using them, talking about those papers specifically and what they found, but also just using them as kind of these avenues for understanding scientific and media literacy, maybe some sort of statistical biases. Maybe, sometimes papers do a good job, sometimes they do a bad job. So just keeping an open mind and thinking about all of this sort of stuff, just logically and skeptically, is what I'm hoping we gain from all of this. Now, with that being said, that's pretty much it for today. I'm very happy to go back to any slides, take any questions. I know I covered a lot. I kind of tried to summarize probably, half a year's worth of statistics and science sort of topics into just 45 minutes or so. And so I'm definitely happy to go back, look at any of the slides again, talk about any of the examples again, or hear any other random questions or comments you may have. So thank you. Let's see, let's open up the chat.
Thank you, Brian. That was wonderful and a lot of information. Very, very interesting. So like Brian said, everyone, I will email out these 2 documents to everyone who's in attendance today. And we do hope that you continue to join us for the rest of the series. I'll also take the recording of today's webinar and send that to you as well so that you can reference it and look back on anything you may need. And if you've got any questions for Brian, now is the time. You can throw those in the chat.
Let's see. Not many questions. Everybody understood Bayesian statistics? You guys are all statisticians now? Awesome. Love
There's
it.
one.
Okay. Let's see. Are there any resources for knowing bad sources? Yeah, so we'll definitely be talking about that as well. We'll talk about that next week and probably in 2 weeks, especially. So the big things are, let's see. So for knowing bad sources, really it comes down to a couple things. Understanding some statistics and all these different sort of logical fallacies. If you understand the things I talked about today and other logical fallacies and things that go with it, you can start to notice when writers kind of make a leap. They say one thing, and then all of a sudden there's this non sequitur, and they say, oh, because this happened, that proves this is going to happen. You see politicians doing that a lot on both sides of the spectrum. They say they quote some data, and then they say, oh, we're going to fix it by doing this. And that doesn't necessarily mean that those two things are correlated. So I'd say the biggest things that you can do to recognize bad data are knowing where the source came from. So who's posting it? Is it posted by somebody with a specific agenda? Is it posted on a kind of a far right or far left wing sort of platform? Is it posted by some random person with a blog at home that doesn't have any actual credentials? Because it's not saying that scientists and people with degrees are not wrong. I'm not saying that everything that's peer reviewed and academically published, that doesn't necessarily make it correct, but it does give some credence to it. So you want to know the source and what credentials that author has for saying it. It helps to see the sample size. If people are making some sort of inferences and trying to make some assertion, is it just on some one little anecdotal piece of evidence, or do they have a huge sample size? If you look at medical sort of data, people, let's say allergens or diseases, things that really affect human health and safety, a lot of those sort of papers, they do these analyses with tens of thousands of people, so many data points, so that the idea is chance kind of gets weeded out and you start to see the actual patterns start to emerge. Let's see. So what other statistical concepts are common in scientific research? Yeah, so there's a lot of different statistics that are used in scientific research. But the core of it is really trying to decide when your observations are better explained by random chance, or if they're actually best explained by your hypothesis. That's really the core of it. And a lot of statistics is kind of centered around getting to the truth and figuring out, okay, let's say I pick a card, from a deck of cards, and you get, I said, the ace of spades, that's our card. What's the chance that happened? What's the chance that maybe this deck is all ace of spades? If we only look at one card, we don't really know what else is in there. So a lot of statistics, I can't cover it all in one talk, but we will definitely get into it in the next few weeks. It's really just about whittling down the options of what's most and least likely. And then somebody also mentioned what p-hacking is. And so I mentioned it here. It's just kind of as a wayside, and we can definitely, I think we'll probably talk about this in one of the future weeks a bit more extensively. P-hacking. So if you kind of take a step back, the p-value is kind of this measure of how sure we are that a statistic is accurate, right? So I'm not a math professor, so I'm not going to necessarily get this all perfectly well explained. But the idea is, let's say you run a bunch of statistics on your data set. Let's say we go outside and we measure the length of some fish, and we go and we measure the length of a whole bunch of this one species of fish, and we get kind of a bell curve for how big that fish is, right? We've measured 100 fish, and they all kind of fit into this general category. Most of them are kind of in the middle. Maybe they're about a foot long. Some of them are a little shorter. Some of them are a little bigger. Now, what we want to do is, let's say we find a new fish, and we're wondering if it's the same species, and we measure it, and it's a bit bigger than this bell curve over here. What's the chance that that's actually the same species, right? And so you get a p-value. You get kind of this measure, this metric for likelihood, how likely it is that it is actually kind of the same as this other data set over here. And p-hacking is when researchers or statisticians, they use ways that are kind of legal mathematically, but you can tweak the data a bit, and you can make it more likely for the data, for the statistics to kind of suit your own interests, let's say. And we'll go into this a lot more later on. But what we can say is that with p-hacking, maybe we take a whole bunch of different metrics, take a whole bunch of different observations, we kind of cherry pick the ones that we like so that the statistics work in our favor. If we're writing a paper, we want all of our statistics to look good and strong and interesting so that it'll get published somewhere. And p-hacking is kind of this method where you kind of massage the data a little bit, intentionally or not intentionally. A lot of p-hacking can be done just because you did bad statistics. But specifically the hacking part is usually it's thought of more intentionally. You run the numbers a few more times, and maybe you run a simulation a few times until the outcome comes out the way you wanted it to. So we'll talk about that a bit more, but that just kind of has to do with misusing this field of statistics to try to support your own argument. Let's see. Next slide. Oh yeah. Boop. So yeah. So here, this is kind of just a quick overview, I'd say, of what I think of as kind of the different steps, the game of those different rungs in this game of telephone of the information filtering down, and all the different potential problems, whether intentional or not, that can make data look different by the time it gets to the end. Let's see. Yeah, I know, definitely, it's tough to not have your eyes glaze over when you're looking at all of the stats, when you're looking at a bunch of numbers and equations. Hopefully we're able to break it down a little bit, with a couple different examples so that it made it a little bit more manageable and understandable, even if you're not going to go out and start applying this equation anytime soon. But if you look at Bayesian statistics now and you see this, and you see a paper that has this equation in there, at least now you've seen it and you kind of know what it's about. And you even know how to use it a little bit with, let's say this example here. Okay, are there any more questions or comments? So next week, what we'll be doing is hopefully whoever wants to join will have had a chance to read these two papers. And then we can come equipped to talk about them, and we'll talk more about other statistical things, other sort of scientific literacy topics, but it'll mostly be focused around the theme of these two papers here.
Wonderful. Well, thank you so much, Brian. You really illustrated that topic well for us, and I look forward to seeing everyone back next week. I will get these papers and a recording of today's webinar to everyone by the end of the day tomorrow. And feel free to reach out to us if you have any further questions. And we will see you guys next Wednesday at 2 o'clock. Thank you.
Awesome.
Thank you, Brian.
Thank you, guys.
Bye all.